Good? Okay, good. Good morning, everybody. It's an honor to be able to open today this session on regulatory and system genomics, and I'll start by maybe a bit giving the context of this research topic. That is to understand the mechanisms that govern regulation in cells. And one of the main topics that remove that. One of the main principles that has been understood in studying this is this interplay between so-called cis-regulatory elements and transacting factors, and this is best exemplified with the act of transcription factors, which are proteins that have DNA binding affinity, and they do so, or sequence-specific transcription factors do so by recognizing literally words or motifs in DNA. And upon binding to these locations can recruit the transcription machinery and transcribe the RNAs. And this controls then the activity and production of messenger RNAs and eventually of proteins of specific loci. So the abundance of those transcription factors in particular conditions will dictate when and where a particular gene will be transcribed. And this duality between the motifs and the transcription factors is also true, or the transacting factors is also true at any steps of gene regulation. You see this for RNA processing, notably for splicing, where RNA binding protein as well as RNAs can bind to elements encoded in the RNA, coming from elements encoded in eventually DNA, to regulate splicing. Same happens for translation, for RNA degradation, and as well in some more complex way with protein degradation. So the goal of regulatory genomics is to understand both the code, but also all these dynamic systems that will dictate what are the concentration of the regulators in particular condition. But within this talk, I want to focus on just the task of inferring the cis-regulatory code. And this I will define it like this, that for a particular gene expression quantity that can be RNA abundance or transcription rate, splicing rate, degradation rate, many others you could name, infer the function which in a given acting factors context or specific abundance that can be determined by cell types, species of interest we're looking at, or the environment. How does a sequence, and eventually any sequence, in this local region, such as a promoter, how does that will lead to a particular wise or a particular quantity? So I'll tell today, this problem can be seen as a machine learning problem if we have enough pairs of y's and x. And my talk is structured on two approaches here. Approaches that have used the supervised learning paradigm, in which you have a lot of labeled data. So you have both the genomes and the genetic sequence and the measurements of these data. And in the second part, I'll talk about semi-supervised, where we have a lot of X, but not necessarily always with the measurement. So the first one is focusing on studying how far we've been in understanding the transcription of regulatory code of humans and mouse with models that have been trained in a multitask fashion using the ENCODE data, using as genetic sequence the reference genomes. And these studies, the study of those models, is something that has been led by Alexander Carlos in my lab in a publication we published it earlier last year. So the idea here is that when such a model is trained on data, we have in the computer an in-silico theory of the regulatory code. And we can confront this theory to experiment. In particular, we can confront it or help it generalize to sequence it has not seen. And here the interesting aspect is to look into artificial sequences. Remember that Informer, the model we've been looking, I'm going to show you results about, has been trained on the reference genome. So it hasn't seen artificial sequences. However, there are many assays where artificial sequences that deviate from the sequence we've seen in the human genome have been proposed, and we can see how good the model fits and what are the principles that have been well captured or not so well captured. So Alex started looking into the proximal elements, so those elements that are in the promoter. And for this, there are many assays, and I'm going to show you results for one of these assays, but in the background, we look at more of them. So this is an MPI study in which transcription factors motif have been inserted or disrupted in promoter elements, and their effect has been measured with a reporter assay. And what you can see on the y-axis is if you replicate this experiment in the computer using Informer, you get a relatively decent performance. So the Pearson correlation is 0.7. It's not perfect, but the large effects are indeed captured quite well. And so this is quite remarkable in the sense that the model has been trained only in sequence from the reference genome. We can even go a bit further. We can use these models to interpret how the predictions are done. I'm showing you here the results for one of these promoters. And in this particular series of experiments, a data box, which is an important motif for transcription, is inserted at several places in the promoter. And what you see here is that, first of all, the predictions are shifted, but mimic quite well what is measured in the assay. So the position of where this data box will have a positive or negative effect on transcription are pretty well captured. But what we found is that if we interpret actually the model and we ask the model at every base what contributes to transcription, which we figured out, or Alex figured out, is that this position here is not a position where data box doesn't work. It's actually a position, we think it's a position where there's a CAT box that is disrupted if you bring in the data box in that position. So there's an interesting thing we can do with these models. So then Alex looked further beyond the score promoter, the close vicinity of the transcription start site, and looked at enhancers, which are regulatory elements that are more distal and that are typical for higher eukaryotes and are a bit less easy to study, but there are also a lot of assays that are probing the biology of enhancer promoter regulation. So there's a lot of results now that go into actually a simple law that seems to say that the expression you see at a gene is the product of the strength of a promoter times the strength of an enhancer. The strength of a promoter is a very complex nonlinear function of the underlying sequence. But once you have that and one of the enhancer, you get a pretty good approximation if you simply do this product, where you just look at one promoter and one enhancer. So Alex asked, is that recapitulated in the Informer? And what he did is he generated sequences of this kind. So he looked at many promoters and artificially put many enhancers. And then compared the product role to what the Informer, which is a very deep model, highly nonlinear model, predicts. What he found is this, that actually Informer learns this simple rule from just looking at natural sequences in the human genome. As you see, there's a bit of saturation, which might be even biological. At some point, promoters get saturated, you can't transcribe too much. And also on the low end, you can't go below zero rate. But otherwise, this rule seems to be captured by the Informer, and it's also in line with many now orthogonal identities. The next question that Alex asked is, what is the effect of distance? So for this, he looked at assays where CRISPR interference studies have perturbed enhancers, and you see how this affects the expression on the target gene. And here we looked at two data sets of Foucault and Gasperini. And in these data, what is reported is that there are long-range effects, so enhancers that affect gene expression even at a distance of 200,000 base pairs. However, the Informer has this very strong decay rule that actually the prediction of an enhancer effect is decaying largely. There may be a bias, and a certain bias in the assays, because what you see here is some kind of positive effects, and it's not necessarily obvious that all enhancers that are far have such strong effects. Nonetheless, the decay here is very strong, and also if we look at, for example, QTL effects in GTEx, what seems to be learned by Informer is a stronger decay than what actually happens So we asked why is that, and then we revisited how the predictions of Informer function. So Informer is using about 200 kb, so-called receptive field, so it looks at 200,000 nucleotides, but what we can do is we can rerun the predictions by just putting hands on flanking regions and reducing the actual sequence we provide to Informer. And what we found is that you get, with just 1/5 of this, the receptive field predictions that are very similar to what you get globally with the full receptive field. So maybe Informers are actually rather learned effect of sequence that are local and could not really capture the effect of the sequence that are far. And this is maybe obvious because proximal elements hold most information. What we're showing you here is the cumulative fraction as function of the distance between the transcription start site and particular regulatory regions of interest. And what we find is that if you look across the genome, there's a kind of almost uniform distribution of things that look like enhancers, but when we look at things that are like EQTL or validated enhancers, they are positioned way closer to the actual transcription start site. So ways of learning these roles is extremely biased to local regions and might be natural that the Informer actually have inherently a strong decay rate for predicted effect of enhancers on promoters. So what we learned from these studies is that this model seems to have learned quite well rules of proximal regulations. They generalize well to perturbations that were not in the training data. There's probably still ways of improving it, but maybe by going to single nucleotide resolution assays, but this seems to be quite well understood. However, this proximal regulation seems to be most of the signal captured by the Informer. When we come to distal regulation, we found that the multiplicative model seems to have been also captured by Informer, but the relation to distance or how to capture very long range interactions is still not done with these models. So to solve that, maybe we need different data. Maybe we need more different models. It can be that the law that dictates how an enhancer touches a promoter is not encoded in the sequence in between, but very far in the folding of entire chromosomes, and then we need different models. Okay, the second part of my talk is about unsupervised or semi-supervised approaches, and this is a work done by Alex again, together with Johannes and Dennis. And here we looked at way more species. We looked at species out of the fungal kingdom, and we move beyond just transcription, but also look into post-transcriptional regulation. The motivation for that is to contribute to this field of representation there. So I'll start with this little quote from Eric Lander, Genome, about the book, hard to read. If you get just this as input sequences, it's going to be indeed just hard to know, to predict your gene expression. And it's how those models are exposed to, right? You just one hot time code the nucleic acids, and then you try to predict encode. With a lot of data like encode and deep learning model, we managed to do that. And this is managed essentially by a neural network, which toward this last layer have identified a representation of the sequence that contains these regulatory elements. So in a way, we're building tools that are able to map this nucleic acid into the feature space that have the features that are important for gene expression. So there are other ways to do that, and unsupervised learning is an alternative to get representations that are also predictive for the output. So that's the idea of semi-supervised approach, is you learn how the sequences distribute in your input data, and hopefully things that are repetitive, that occur more frequently than usual, happens to be also those features that are important for your prediction tasks. And luckily, maybe in biology, that's a bit what happens with evolution. So functional elements, elements that are actually in interactions with our DNA binding and RNA binding proteins, transcription factors, RNA binding protein, microRNAs, they have a footprint. They can be, depending on the context, selected positively or negatively. And so they provide us features that we distinguish the distribution of sequences that we see across species from uniform sequences. So if we learn representations of X of how the genome sequences distribute across species, maybe we have features that are helpful for downstream tasks. So in a way, we are coming back to comparative genomics, but with new tools. Comparative genomics has a whole history, and one of the workhorse algorithms for that has been sequence alignment. But sequence alignment, when it comes to regulatory features, is a bit limited. And I'm going to show you this here with untranslated regions, starting from Saccharomyces cerevisiae, the wicked yeast, as a core species. And what we show you here is how the coding sequence is aligned using a simple blast alignment across other species, across other fungal species, and how UTRs align. And so very quickly, you will not be able to align your UTRs, but you're still able to align the CDS. So that shows that this alignment worked well for proteins, but it's not so good for regulation. On the right-hand side, I'll show you here now that about 200 nucleotides, which are downstream of the stop codon of CBP3, a protein in cerevisiae, and it's a protein, it's a UTR that contains the motif for an RNA-binding protein that is conserved all the way to point A, and that's called PAF3. And what we find is the motif is actually found in all those downstream regions. There are even experimental evidence that it shows it's functional in CASA. It is not clear, but maybe it is also functional in all these other places. You see the element is mobile, it's not always at the same place. These sequences don't align very quickly, but the motif is there. So maybe with alternative algorithms, we can recover those motifs. And that's what we've been investigated. We said, let's use algorithm from natural language processing that learn ways of representing sequences with the trick of masking parts of the input. So in our case, we are masking nucleotides, and we ask the algorithm to recover the nucleotide that is masked. And the idea is that the representation that those algorithms capture, hopefully understand words and context in which those words are important. So we've done that, and the difference compared to other DNA language models that have been published also over the last years is that here we go very far in evolution. We go so far that the regulatory code is evolving. So we knew that if the regulatory code is evolving, you need to have the model, and so we give to the model the species as a covariate, it counts as a token inside it at the start of the input sequence. And so the model is learning not only the elements, but in a species-specific fashion. We worked with fungal species with more than 1,000 genomes and 800 species. And we have done a held-out experiment where we remove an entire genus, the genus of Saccharomyces cerevisiae, all the species out, so that we don't learn by heart the Saccharomyces cerevisiae genome, and then we will do a lot of test experiments in that genome. What we've trained, what we can do in these genomes, we know that we can align well coding sequences. So we start with the coding sequences as an anchor, and then we extract the regions which extract the nucleotide downstream and 1,000 nucleotide upstream. So these regions typically for fungi cover more or less the 3' UTR, whereas this region contains the 5' UTR, and the promoters sometimes go out to the upstream gene. These have 10 billion bases, and these have 3 billion bases. So the first question we ask is, what happens if you look at a particular randomly picked position within one of these regions? I'm going to show you the results for the 3' UTR. And what we found is that these models, these very fancy models, don't do much better than a model that just does the majority class, or models that just look at a few k-mers. And the reason is that in UTRs, most of the regions are simply driven by dinucleotide sequences that have no motifs if you look at a random position. It's actually what you expect. So looking at just these loss is not so informative, but then we ask what happens if we look at the motif. And in cerevisiae, the motifs are well known, and now the picture looks differently. This species-specific language model reconstruct much better those motifs than random sequences. And even for some region, it's just as good as an alignment among the genus. this is an enlarged species, the antigenous was out. So that's interesting, it tells us it's recognizing those places. So if I come back to this example of CBP3 3' region, this was my pictogram at the beginning. What you see here is the reconstruction likelihood of the correct base, and we have within those boundaries higher likelihood of the right nucleotide. So this gives an indication that indeed these motifs are recognizing the regulatory elements. We went a bit beyond, so we asked, since we look at so many species, the regulatory code can evolve, and this is an example with 1.1p. So this is the transcription factors which is present in some species, but not all in our data set. And so we asked, can you recognize this motif? And it does so. But the interesting thing is it does so better for the species that have, when we blast RAP1, that has 1 point in the genome compared to species that don't have it. It's not true for all, but it's a signal that the motifs that are recognized indeed are sensitive to what the genome encodes, even though we didn't tell about protein content to the model. It's just an indicator of the species. We next ask, does this model get a sense of context? This box we put there is a broad box that contains UTRs and promoter. We don't tell the model where the UTRs are, but we know this for Cerevisiae, this has all species. So we can ask, is there a sense of that? And the primary thing we did is just look at nucleotide predictions. And what you see here is the actual and the predicted for the A and for the T in blue. And there's a pretty good agreement between the preferences for the nucleotide with respect to the TSS, this position here, 0, which was not given to the model. The model at the start codon, but not the TSS as an anchor point. You can ask then, is that also helpful for motif reconstruction? And so we have similar results also on the 3' UTR. I'm going to move again to 3' UTR for this. We ask this +3 motif that you can recognize. You recognize it better if you're inside the RNA, so within the 3' UTR or outside. The box is bigger, so we have many places where there is actually the polyesite in between, and so we can find instances where this motif is actually present in the DNA, but outside the mRNA. And we ask the model whether the reconstruction is different, and it does so. So there are a few here instances of this motif, and the way it's reconstructed, which is shown with this motif, is very correct and strong before the TSS, and then it's not actually reconstructed when it's transcribed, sorry, the polyesite, and it's not reconstructed after the polyesite. So that's strong indications that this model is understanding context. And yet another one now, this is the preference of the Tata box. Cerevisiae and Pombe have different mechanisms in transcription start site, and the functional Tata box in Cerevisiae can act very broadly within the promoter. In Pombe, it's probably peaked around 35 base pair before the TSS. And so same, we asked the model, can you reconstruct your Tata box? And how do you do it as a function of the position in the gene? The TSS was not known. And again, if you align things by the TSS, the model, the species over model, reconstructs the Pombe Tata box very precisely at that place. So you trust this compared to other places. So again, the motif exists, but the model reconstructs it strongly if it's at the right place. So this also indicates that this model kind of understood where promoters are, how elements of the promoters should function. So we can then move to the downstream goal, which is, can I use now these representations for downstream tasks? And in doing so, what we've done is we simply fix the embedding. We don't retry the model. We just take the 4 last layers and do a linear regression or ridge regression on this for several downstream tasks. I'll show you one that is non-trivial. This is to predict the effect of perturbation of the data. So here, this is data from the lab of Iran Segal, where in a Titan fashion, we shuffle sequences in a 10 base pair window for 200 UTRs in a Cerevisiae. And what we find, this is one example, and this is for the overall library. The model predicts pretty well the effect of those perturbations. Again, it's just a linear regression made on those embeddings. So that's quite encouraging, and we've done this for many tasks. gene expression for COMB and RegSys for RNA half-life, and PRA gene expression and PRAs. The performance varies between those tasks, but we always get a model that is better than state-of-the-art models or naive approaches based on k-mesh regressions when there's no state-of-the-art models. So in conclusions, I hope I could convince you that DNA language model can capture conservation in an alignment-free fashion, that species-aware DNA language model can capture the evolution of the regulatory code, and that this technique allows us to have representations of sequence that can be helpful for predicting gene expression-related traits. We think that these tools can help in comparative genomics to study under-species genomes. So in the future, we hope to be able to apply this to more species and indeed contribute in understanding gene expression for way more species than what have been studied so far. And maybe more on the theoretical aspect to revisit the notion of conservation without being based on alignment. With that, I'd like to thank you for your attention and thank the people that have been working on this work and name them along the way. But you can come to the posters, as mentioned here. You can see the preprint of the species-aware DNA language model here on bioRxiv. Also from our lab, we have Laura Martens, who has a talk today at HeatSeq on single-cell at-axis count. And I also have another talk on RNA splicing and rare disease tomorrow on the RNA coding. And if you are looking for a position in the topic, we'll be welcoming. Thank you for your attention.
So if I understood correctly, you showed that the informal model fails to predict long-range interactions and the effect of very long sequences. How do you think that we can resolve this? Is it just that we need more data on very long sequences, or the other ways of computation we can face this challenge?
We're not working on this. So let's see how people manage this. So it can be that you need more data because the physics, as I said, that will be my guess now, is that the physics of them bringing those loci together is more global, is encoding the entire chromosome. And so we need to model that. There is a deep learning model called, I think, ORCA that works on this. I don't know any fusion of ORCA with Informer. Probably people work on that. Maybe this address the problems. Or maybe we need more data of different kinds. ORCA is trained on IC, so it does also look into it. Yes.
So let's see or again, put that up. Anyway, great talk. Lots of questions, but I'll ask one. So can you say a little bit about? So you use mask language models. I was curious, like, there are other models, just like a sequence variation model encoder. Why is the masking actually important? Can't you look like, have you compared to say regular sequence variation model encoder like E, for example, which is trying to predict variants?
So we have not explored all the possibilities of doing this. In this study, we've rather asked, is it important to bring to this model the variable species, the species identity? And that's what we've tried, only with mass language models approaches. But with different architecture. I showed one that is based on a transformer, on DNA bird transformer. There's another architecture that we've used also in the past. We haven't been into versional approaches. They may have add values, yes, but
Why is the supervision important for advanced understanding? Why is the
it's convenient. I don't know if it's important, but it's a convenient way to transforming the problem from learning a distribution, from learning conditional distribution. And this you see in many aspects of machine learning. This is the concept of self-supervised learning. At the end of the day, we're not necessarily want a representation a model of the distribution, we want a representation. So if this training conditional distribution is enough to get good representation, we've done the job. And that's easy because it's back to supervised problem. Does that answer it? Not really, but we can discuss it.
Hey, over here. Great talk. Thank you very much. So I have a question on your second part, basically following up on this. Are you using just atomic symbols in a way like A, C, G, and T, or are you using also more complex motives? Because, I mean, natural language processing, you have something like bypair encoding.
Yes, so
you can enrich your alphabet.
Well, don't play too much with that. So, in our early studies, we were just having, the token was the nucleotide, and we masked the nucleotide. Then eventually we just took what others have done so far, which is so this DNA bird turned out to perform better. We didn't dissect why it performs better. What DNA bird does, it looks at 6-mers. The 6-mers are overlapping, so doing this masking is a bit tricky. When you mask one nucleotide, you need to take out anything that overlaps it. It is thought possible, and that's what we do. We have not thoroughly tested whether going with the 6-mers has any added value. I would also like to get rid of that if one can show it. So we've been pragmatic and just ask, okay, adding species help, yes, no, and that's what we've done in that study.
Thank you.
Hi, I'm sorry, I'm on the floor here.
Yes.
So I was wondering, so in your species transfer, you're working with yeast and they don't have entrance, right? For the most part. I was just wondering if you are working on how in mammalian systems, when you have entrance of regulatory elements in the entrance, if you have tried how your model extends to those situations.
Not yet. So the Cerevisiae has few introns. Other yeasts have more introns, but there is no enhancers as far as I know. So this overlap is not there. But yes, of course, we also have a lot of work on splicing, and we're interested in looking at those regions. And the same concept, taking an exon as an anchor and then looking in the vicinity could be hopefully working.
Thank you, Julien. Really nice talk. I was wondering about how much sequence diversity or evolutionary distance you need to learn these models. So how many species are far away in terms of evolution do you require to be able to learn something that is meaningful? Because I mean, if the sequences are too similar to each other, then you will not be able to learn anything, right? You learn the differences, right?
Yes, I think this is a key question. We have not explored that. So indeed, what you do with this, with those approaches, you're sampling observations from an underlying distribution, and the question is, what is the distribution you're after? Yeah, and exactly as you say, if you go too close, one can do the same just among human genomes, and then we'll learn. I'm not sure what we learn there, but like very small variations. So going into further away is good. If you go very, very far, all the way to prokaryotes, is that helpful? That's unclear. So I think most of the work is actually not so much on the architectures of the models, but rather in defining clearly what do I sample from and for what purpose, and how do I evaluate that. We need metrics. We don't have metrics yet. But yes, this is very important. Thank you.
Time for one last question. Anyone has one? Okay, I have one. Maybe this is already asked in another way, but do you think these will extend to mammalian systems where you have just a lot more unstructured sequence, out to distal enhancer elements and things like that?
Yeah, so people do that. So there's a lot of DNA language model on
reconstruction in the way that you did it, to reconstruct these elements when you don't know where they are.
People have not studied it as we did in details what is in the belly of those models, and I don't know what is it in those that have been tried on humans' entire genomes. I do think that the angle we took, which was to focus on aspects where we know what to expect, like these core UTRs and co-promoters, might be also helpful to have more, let's say, consistent representation. But it's all good.
All right. Okay. Let's thank our keynote again. Thank you.